This conference will now be recorded.
All right, folks. So it's nice to see you all here tonight. Thank you for coming. I appreciate you attending, and I also appreciate the kind words that you've said to me regarding with me recuperating and feeling better, as well as with my service. And thank you so much. Really, it means a lot to me. And truly, my students mean a lot to me. You all and my other classes and such. I care about what we do and how we share this material with you all in order to learn and move on from this class to the next class and moving on to your prospective schools that you would like to choose in order to work with patients and such. So it's pretty exciting stuff. So I'm glad you're here. So tonight we're going to be looking at the muscular system and we've already been reviewing the videos and reviewing muscles in lab. So this Monday was a very productive evening. I hope that you felt it was that and that we were all able to take a look at and do videos of all of the muscles that you are responsible for the lab, as far as for your practical number 2. So we've really we accomplished a lot. It was a very busy night, but I'm really pleased that we were able to get all that done. And so this coming up week, we're going to be focusing on and looking at. We'll be doing a review as far as for the musculature, as well as I believe that. And I have to confirm as far as when we're doing our testing and such like that. So sorry, I'm not remembering off the top of my head, but I'll respond back to you all regarding what we're going to be doing as far as lab work and such. But primarily it would be reviewing the musculature and those models. And please, I would highly recommend you actually touch the model, you actually looking at it, saying the terms out loud as you're looking at the different pieces and such. And don't get overwhelmed, get motivated, right? And if you need any help, I'm here to help, please. Okay. So we've already gone over these terms, and these terms were located in my first note packet here. You'll see excitability, contractility, extensibility, and elasticity. Right, the excitability, the ability for muscle to be able to respond to the nervous system and particular action potentials that would be generated and sent to stimulate the neuromuscular junction, which in turn, well, really at that neuromuscular junction, we have its chemical synapse, and acetylcholine is going to be released within the synaptic cleft, and we're going to then create a situation where that acetylcholine will travel across the synaptic cleft to specific ligand-gated channels. They're ion channels that will respond. The acetylcholine acts as a key to unlock the gates, those gated channels, and they'll open, and then we're going to have ions moving back and forth, and we'll talk about this in a few minutes. And it'll release it'll really begin the process of an action potential being created at the neuromuscular junction, right at the area of the motor end plate, and it'll travel across the sarcolemma and begin events that will take place in order to eventually lead to muscle contraction. So pretty exciting stuff, pretty neat stuff, pretty challenging information, to be honest, and you know that. But you all have been doing your part as far as transcribing the videos and writing down those notes from those videos so that you know step by step what's taking place at the neuromuscular junction, at excitation contraction coupling, and at the ability for having the cross bridges occur between myosin and actin, and then allowing for the isotonic contractions to take place. So, and we're going to talk about isometric and isotonic this evening here. Okay. So we've looked at these as far as the excitability, contractility to be able to shorten forcibly, right, when stimulated, right, that ability to be stimulated. Extensibility and elasticity work in conjunction with each other. So we have muscle has the ability to stretch, and then ability to also, as far as elasticity is concerned, to recoil back to its original starting position. So we've gone over that. So let's go. All Very good. Now, the neuroelectrical factors that are taking place here, right? So one of the key issues that I want you to recall and know, and I'm going to really go to the notes for this, but I'm going to show you some images in the PowerPoint regarding this. But please, you should work in conjunction with your PowerPoint and your notes with your studies, as well as, reviewing the chapter and reading through it at least once, right? But know that really sodium ions are moving first. Okay, sodium ions are always moving first, and then potassium ions follow. That's a key crucial issue that you need to keep in mind. And you'll see here this resting potential, or I'll refer to it in notes as the resting membrane potential. Okay, we're going to see, and let's look right here. So we're going to see that really there are sodium and potassium ions on both sides of the sarcolemma. Realize that. But you need to remember that and look at what it says up here, that the sodium ion concentration is higher just outside of the plasma membrane. It would be in this region here. And potassium ions are higher in concentration inside the cell. Okay, so outside, higher concentration of sodium ions, inside, higher concentration of potassium ions, but know that there are sodium potassium ions on both sides of the sarcolemma, the plasma membrane of the muscle fiber, muscle cell. Okay, and this is, we would say that the positive on the outside, negative on the inside, that represents the resting membrane potential. That will change when an action potential is generated and travels down the sarcolemma. Okay, so that's important for you to remember that and keep that in mind. But knowing that resting membrane potential, positive on the outside, negative on the inside, close to the plasma membrane, and this would be in the case of the sarcolemma, it's the plasma membrane of the muscle fiber, muscle cell. That's what you need to keep in mind. Now, I'm going to go to my note packet here. So here's muscular system notes, part 2. So the electrical events that take place at the sarcolemma, okay, we're not talking about the motor neuron, okay, we're not talking about the motor neuron that's going to be having an interaction between the nerve as far as the axon terminal and the motor end plate. We're not talking about that. We're talking about right now what occurs at the motor end plate, okay, on the muscle side of things, not on the nervous system side, but on the muscle side of things. And we're going to say that at the area where the neuromuscular junction is located, the motor end plate is where we have the acetylcholine passing across the synaptic cleft, that space at the synapse, the chemical synapse, and we're having then those ligands, the ligand, the acetylcholine, that neurotransmitter, is going to engage with the specific receptors on the motor end plate. Okay. Depolarization, there'll be a local event, local depolarization at the motor end plate, and then in turn it will turn into a depolarization event that will travel down the plasma membrane of the muscle fiber, the sarcolemma. And then in turn, we know that once there's a depolarization, we need to return to the resting membrane potential, so then there is a repolarization that needs to take place. So it's always depolarization, repolarization, depolarization, repolarization. And that repolarization will allow us to return to this resting membrane potential. Now, the measurement of this, as far as volts are concerned, would be -70 millivolts, okay? And again, higher concentration, remember these brackets, concentration, so higher concentration of sodium ions outside, higher concentration of potassium ions inside. That's the resting membrane potential. We started here, we end with it here. Understood? Okay. So that local depolarization, I said to you that the acetylcholine binds to the specific receptors at the motor end plate. Acetylcholine is the key to unlock those ligand-gated channels, right? And they're ligand-gated ion channels, actually specific ligand-gated, and I'm going to correct this here. You can add information here, but ligand-gated. Ligand-gated cation channels, meaning that these are channels that are positively charged. The positively charged ions are passing through these channels. Now, sodium diffuses into the muscle fiber. So sodium moves from high concentration to low, right? I'm going to add this here. So movement from high concentration to low concentration. Actually, I should put them in the brackets. I think that's a good idea. That and that. Not good. Okay. Sodium ions are moving first from high concentration to low concentration, from outside to inside, right? The interior of the sarcolemma becomes less negative as a result of more of these sodium ions rushing in. This will result in a local event right at the motor end plate. From there, we're going to have we're reaching a certain threshold, a certain number, okay? So it's going to become less than, so if we have negative 70 millivolts, it starts going more towards the positive, this number right here, as far as the resting membrane potential changes because there's more sodium ions moving in. So then depolarization is when the motor end plate reaches this threshold, this number, this value that is more positive. Depolarization of the sarcolemma will propagate down the entire sarcolemma. So it spreads from the motor end plate, it spreads along the sarcolemma. And what will happen when it does this is then, as we know, when we were watching the videos and going over what takes place, then this is going to initiate the events that will release calcium ions into the sarcoplasm, which will in turn lead to the cross bridges taking place and muscle contraction occurring. Right? So the motor end plate reaches a threshold potential, threshold value. It's going to be more positive. The depolarization of the sarcolemma will propagate down the entire sarcolemma. So this threshold, the motor end plate potential, reaches a critical level, a certain value, certain number. It's more positive. More positive than the resting membrane potential of negative 70 millivolts. So it's going to be a value that's more positive. The sodium ions are going to then rush inward through voltage gated channels, because again, now we're creating this electrical event on the sarcolemma. So voltage gated channels will open, allowing more sodium to rush in. An action potential is produced and will continue away from the area of the initial depolarization. Action potential spreads from one section of the sarcolemma to the next, and then what will occur is then this will contribute to then calcium being released into the sarcoplasm, and then the cross bridges will occur and contraction will take place. Now, repolarization means that we're going to actually then return to that resting membrane potential. So we have to have other channels opening. So what's going to happen? Well, sodium voltage gated channels will close. Remember, they were open. Sodium is always open first. Then potassium gated channels will open, and potassium will then move from move outward. There we go. Yes, we will move outward because they're going to move from a higher concentration inside to outside. So they move from in to out. So they're going to move outward from the muscle fiber, and this will allow for then that resting membrane potential to occur, where we'll be at approximately negative 70 millivolts, right, as the movement of that potassium ions. Now, also know that ion levels in and outside of the muscle fiber, they're restored by what's called the sodium-potassium pump. The sodium-potassium pump, it's an active transport. So if we know that passive transport, let's go back to our image here. Passive transport means that if we have higher concentration of sodium ions outside of the cell, and there's a lesser concentration of sodium ions inside of the cell, movement from high to low, right? We're going to be moving sodium ions just through channels. They'll just be able to pass from high concentration to low. It's diffusion from high to low. We're just going to be moving from outside of the cell to inside of the cell as far as sodium ions are concerned. Sodium will move outside to in. Now, When those voltage gated channels close, and we're not going to have this passive movement, it's just a passive movement, right? We're not using energy for this. And then potassium ions will move when the voltage gated channels open from inside to out, because there's a higher concentration inside than out. So it just passively diffuses from in to out. I said that before. Now, a contributing factor to this resting membrane potential, the return of it, will be also active transport. Active transport means it goes against, it will need ATP, the process, in order to push ions against their concentration gradient, okay? Meaning that we'll push sodium ions from inside to out, that's against the concentration gradient. We'll push potassium ions from outside to in against the concentration gradient, and we'll need ATP in order to do that, all right? So let me go out of here. So the sodium potassium pump actively transports, ATP is required, actively transport, this here, ATP required against the concentration gradient. So 3 sodium ions will move outward, 2 potassium ions will move inward. And this will contribute to the return to the resting membrane potential of negative 70 millivolts. Okay. Now, I'm going to stop sharing for a moment. Cation, I believe, Norma, I believe cation is one word. All right, so I know, like, in a very short period of time, I've said a lot of information. And so again, rewatching this video and asking me questions when I see you on Monday of next week would be a wise idea, but you can ask some questions now if you would like.
I have a question.
Sure.
So depolarization does not need ATP.
Well, depolarization, really, it's a matter of that we've already, yeah, you're correct in that we don't need the active transport just then. Really, it's at
repolarization uses the sodium pump.
We'll have a combination of the passive transport of potassium ions moving through the voltage gated channel and active transport via the sodium potassium pump.
Okay.
Okay. Mm-hmm. Yeah, this is something too. I'm going to be sharing some videos with you all. There are 3 more videos that I'm going to be sharing with you, and these deal with, in particular, for our next chapter, dealing with the basics of the nervous system and how it functions, and talking a little bit more and just giving you some more information added to what you're already learning regarding depolarization and repolarization. Okay, so let me return back. Go ahead. Sorry.
Could you once again tell me very quickly passive transportation?
So passive transport. So listen to this here. So we're going to move down the concentration gradient from high concentration to low concentration. High to low. That's diffusion. Do you remember that? Diffusion. If I were to open up a cologne bottle, right? So I have this cologne, it's right by me. Smells good. And I open it up. In the room, eventually, the smell of this cologne will diffuse throughout the room so that somebody at the other end of the room will eventually smell this cologne. We went from high concentration in the bottle of the cologne to low concentration out throughout the rest of the room. Okay, so that's diffusion. Movement from high to low concentration. No energy required. It's a passive transport. If I need ATP, then it would be an active transport. Okay, this is why we talk about these things, active and passive transport, earlier on. So passive transport, movement from high concentration to low concentration. No energy required. So when we look at, so let me share my screen again. When we look, cancel. When we look at this image here of the resting membrane potential, positive on the outside, negative on the inside, sodium ions are higher concentration outside, they're lower concentration inside. So passive transport means that sodium ions will move from outside of the membrane to inside of the membrane. Potassium ions are higher concentration inside, lower concentration outside. So they're going to move from inside to outside. Those are passive processes, just moving down the concentration gradient. If we want to go against that, we need energy to go against that. And that's where ATP comes into the picture here.
One more question.
Sure, go for it.
Is this what is happening at the sarcomere when the A band and Z band, like when it's the thin and thick filaments are going in and out? This is what we're looking at.
So what you're thinking, what you have to realize is that when the action potential is generated and we end up having that electrical event travel down the plasma membrane of the muscle fiber, then what's going to happen? Calcium, remember, calcium is released into the sarcoplasm. It's the cytoplasm of the muscle cell. So calcium is released, and what does calcium do? Well, at the area where we have the actin and the myosin, calcium interacts with the troponin tropomyosin complex, does it not? Calcium interacts with that, and it causes that movement to take place, exposing the sites that are binding sites on actin for myosin, right? We have that cross bridge occur, and then we have then the movement, the sliding. That's why we have then the muscle fiber. It's hard to, sorry. Here we go. The muscle fiber, it shortens, the muscle fiber short because we have the sliding, right? It's called the sliding filament theory, right? So good job, Nina. Yeah, that's correct.
It actually makes sense now. Thank you.
Good. Yeah. I know it's not an easy concept, and it's something that you have to review and go over. But once you get it, it's like, okay, this makes sense. But it takes a bit of time. I understand that. Absolutely. So let me move on. Okay. All right. So all right. So what I'd like to do now is I'd like to continue on here and look at this. We're going to just finish up with the notes, this part 2 of the notes. So whole muscle contraction. So contraction produces muscle tension, the force exerted on the load or object to be moved. So here's the thing, and know this, that depending upon what the load, so say I'm holding this paper towels and I'm going to pick it up and remove it, or I'm going to pick up my phone. My phone's a little bit heavier than the paper towels. Well, when I pick up the paper towels, I won't have as many muscle fibers working, right, in order to pick up that paper towel. But when I pick up my phone, it's a little bit heavier, or if I pick up even something heavier than my phone, I'm incorporating more muscle fibers involved in that contraction process in order to lift the load, okay? But there are times when, depending upon how heavy the load is, it can be so heavy that I really can't pick it up with one arm. And so even no matter what the muscle fibers, engaging all of my muscle fibers, I still, because the load is too heavy, I still can't pick it up with one arm, okay? This is just a review of the terms that I've mentioned multiple times: isometric and isotonic contraction. Isometric contraction, and I'm going to put this next to it here. Let's see. And I'm going to put in brackets that cross bridges are formed, but sliding of actin, which is the thin muscle fiber, does not does not occur, right? So so isometric contraction, no shortening. There are cross bridges that occur, so we've got the tightness of the musculature, but we're not having the sliding taking place yet. But isotonic contraction, this is when we have then the shortening, the sliding is taking place, and this is what you think of when you think of muscle isotonic. When you think of a muscle contraction, this is what you're thinking of. classic muscle contraction. That's what you're thinking when you think of, all right, that's what you're thinking of classic muscle contraction. You're thinking of isotonic contraction, okay? All right, now, moving on here. So each muscle is innervated by at least one motor nerve. So this motor neuron has axons that are being released and sent out to the neuromuscular junctions. And so we have the branching of these axon terminals going to the neuromuscular junctions of multiple muscle fibers, okay? And so a motor neuron and all the muscle fibers that are being supplied by it would be called a motor unit, okay? Muscle twitch, the simplest contraction resulting from muscle fibers' response to a single action potential from a motor neuron. So it's not a sustained contraction, okay? Not a sustained contraction. Let me close my email for you folks, sorry. But coming up, let's close that window. Very good, okay? So muscle twitch, you know, and we know, and we've all experienced too, like twitches that can take place even as a result of like some type of virus that can affect involuntary. Yes, correct, good, very good, Nina. That's correct, involuntary. Okay. Now muscle contracts faster than it relaxes, know that. It's smooth and the strength varies with the need, aka also the need/demand, okay? So yes, we can have quick contraction. So hence, and we're going to be talking about this, that regarding reflexes. So if I touch something hot, right away, even without my brain, my cerebral cortex consciously thinking about it, I touch something hot, right away, a reflex will pull my hand away from the hot item that I'm touching, okay? And so what's taking place is that this is very fast contraction. There is myelination of the axons of those neurons that are the muscle neurons that are sending that information to the skeletal muscle to move very quickly away, and then relaxation can occur a little bit more slower than the contraction itself. Okay. This is interesting as far as muscle size is concerned. And I want you to know, and I'm going to scoot down here for a moment and show you one thing here before I come back to that. Muscle hypertrophy, okay? When muscles get bigger, when they get larger, it's not as a result of the number of fibers increasing, but the fiber size itself. So there are more mitochondria, there's more larger cytoplasm, the fibers themselves get bigger, not the amount of fibers, okay? So I want you to know that there. So remember, it's not the number of fibers, it's the fibers themselves that get larger, okay? And really what you're gonna see here is that this is a very good image here. We're seeing slow and fast twitch muscle fibers present within a muscle, and there's a combination of them. So we all have the ability for very quick, short bursts of ability for our skeletal muscles to contract and do certain tasks. So fast twitch would be anaerobic metabolism for fuel, provides a short burst of speed, fires rapidly, fatigues more quickly, sprinting, okay? How about for something like a marathon or a jogging, slow twitch muscle fibers, right? They'll need oxygen for fuel. So we would say it's aerobic and offers extended muscle contraction, fires slowly, higher endurance, great for marathons. So we all have a combination of these different types of slow and fast twitch muscle fibers. But some of us naturally, genetically, have more of one than the other. And also via training, we can create these situations where we can have, whether we're training more for speed or we're training more for endurance, we can also enable that to have more of a, as a result of training, the ability to do more endurance type activities or do more activities that require fast bursts of very quick energy and muscle contraction, okay? That's through training. We'll come to that at the end of the presentation here, but let's see here. Okay, so we've gone here. All right, so we were on muscle size here. So remember, again, it's not the increase in the number of muscle fibers themselves, but an increase in the fiber size that increases muscle size. So muscle hypertrophy, muscles get bigger. That's as a result of training, and that's as a result of the muscle fibers themselves increasing in size, not in number. Okay. Now, as far as contraction is concerned, this is quite interesting. So think of the extraocular eye muscles. We didn't review these in lab, but we will next week, okay? And they're very easy as far as we have rectus muscles that will actually be superior, inferior, lateral, and medial rectus muscles. And then we have oblique muscles that are superior oblique and inferior oblique eye muscles. And these are muscles that will be able to, we can consciously contract them, we could have voluntary control over them so that we don't have to move our head, and yet our eyes are moving in all different directions as a result of the conscious skeletal muscle contractions. Now, these are very tiny muscles, right? They're rapid and brief as far as their contractions are concerned. Think of your calf muscles, the gastrocnemius, soleus. They contract more slowly, but they can hold the contraction longer. They're larger muscles than, say, these tiny muscles of the eye, okay? And the term muscle tone, right? We all have some kind of an idea regarding muscle tone. It's a constant state of slight contraction of these muscles that help to support the body, okay? So understand that if we didn't have this muscle tone of the musculature that supports our body and our trunk and our head and our spine, we would just be kind of like slumped over, right? Because there needs to always be a slight contracted state of these muscles that will help to keep us upright, okay? ATP, I mentioned to you this earlier there, and folks know that we talked about, I know I've mentioned this, as far as rigor mortis. That way, cramp, white. Sorry, Nina, let's see.
Those muscles that are consistently being slightly contracted, like you said,
yes.
The neck muscles, is that why sometimes they cramp up or cause like more tension there because they're always being slightly contracted, or that
No,
is just
it's more having to do with like ions present in different, so that really can be an issue. Yeah.
Okay.
You know, making sure that different ion concentrations of sodium, potassium, magnesium, calcium, that can affect.
There's more concentration there.
Yes, so that can be affecting that cramping and such. Yeah. So I mentioned to you regarding rigor mortis, and this is a state of contracture that occurs upon death. Okay, and what will happen is that we'll have cross bridges that will be created. Okay, but we can't release the cross bridge because no ATP is being produced in order to do that. Right. So as such, we're going to have a sustained contraction of musculature, stiffness, hence the term that's not very politely used term, talking about a cadaver would be a stiff, right? The stiffness that occurs, and only over a period of time, less than 24 hours, it'll break down, the tissues will break down, and the cross bridges will separate, and there will no longer be this contracture that's taking place. So ATP is important for muscle contraction, right? ATP supplies the energy needed for muscle contraction. The only source of energy that needs to be regenerated quickly. Now, there are different sources of this ATP production, okay? And I'm going to show you also, as I talk about this for a moment, then I'm going to show you this image here, which shows you all three of the processes that are occurring. So if you'll see here that via direct phosphorylation of ADP, so ATP has 3 phosphates. ADP has what? 2 phosphates, okay? AMP has adenosine monophosphate that has 1 phosphate. So ADP, 2 phosphates, interacted with creatine phosphate there will produce 1 ATP, okay? And it's a quick process, but it doesn't last very long. An anaerobic pathway, okay, we're not using oxygen for this pathway. Glycolysis, lactic acid formation, 2 ATPs are the net gain. Aerobic respiration, fully taking advantage of oxygen present within the pathway and the cycle, 32, up to 32 ATPs can be produced. So anaerobic respiration yields only 5% as much of the ATP as aerobic, but produces it pretty quickly. But again, too, we can't have sustained anaerobic activities occur. We need aerobic respiration in order to produce the amount of ATP that's necessary, okay? So let me just show you this image here. So here we have the first as far as creatine, and you'll see here that just this pathway, you don't have to memorize this. You do not have to memorize this, but you should know this right here because you're getting an understanding of that. Okay, direct phosphorylation, only producing 1 ATP. Anaerobic, producing approximately 2 as far as the net gain. And aerobic itself, producing very much more, okay? But know that the anaerobic pathway is quicker, but yields much less as the aerobic respiration. And so you're seeing here anaerobic, but aerobic, yeah, absolutely, right? So we have here as far as many ATP are being produced. And I want to bring you back to this term right here, muscle fatigue. So physiological inability to contract despite continued stimulation, okay? And this is something as a result of a depletion of ions.
Like in
Okay,
space?
Pardon me?
Like if you were in space?
No. Just a depletion of ions as far as using them and not replacing as far as nutrition-wise and such. Yeah, and so this also can take place as far as in pathology, where we're not actually maintaining ion concentrations in a homeostatic fashion, right? Homeostasis, no matter what value that we're looking to maintain proper numbers of, whether it's calcium ion concentration, sodium ion concentration, potassium ion concentration, anything, magnesium. The body does what it does in order to do this and to maintain this, right? As far as homeostasis is concerned. If there's an issue, if there is some type of disorder, then we can have issues where, say, in the case of certain endocrine disorders, we're going to have, and really the endocrine system can really help to manage ion concentrations and such. And so using the urinary system, using the digestive system. So, if there's a disorder, then we're not going to have the proper levels of ions, and this can also lead to muscle fatigue. Okay.
Dr. Perrin,
Yes,
is this
go ahead.
anything when someone gets STEM, like if they're in physical therapy and they get the electrical STEM, is that to bring up the ions, or is that something
No, it's
completely
not.
different
No.
related?
Yeah, so really, and I used muscle stim and TENS units quite a bit in my practice because I felt that one, a TENS unit is nice in that it can block messages in the nervous system in order to help with pain management without having to take medications. So that's a positive. That's a good thing. And electric stimulation, so muscle stim, really that's a matter of really trying to strengthen the musculature. So it has nothing to do with the ions now. It has more to do with just trying to, from an outside source, extrinsically create muscle contractions as a result. So it's really, the muscle stim is acting like the nervous system, understood? Stimulating muscles to contract and not just doing it from within. Make sense?
Thank you. Yes.
Yeah, you're welcome. Mm-hmm. Right. So, I mentioned, I showed you this earlier there. So I just want to show you here as far as this is also giving you information regarding the fibers are concerned, as far as then the slow and fast twitch fibers. And know that the oxidative, right, this is for sprinting, walking, whereas the glycolytic, more for intense or powerful movements like hitting a baseball, lifting a heavy weight. This would be fast glycolytic, and slow oxidative would be for more endurance type activities. So you need to know just the names of these different types of muscle fibers and what situation that they might be present within in order to help in a certain type of activity. Okay, and you'll see here as far as we've given you the slow and the fast twitch right here. Okay. All right, very good with that. I'm going to minimize my notes here, and I'm going to go back to PowerPoint here. Okay, let's see. So we've gone over and looked at, and so here, this is a nice image. This is cool, and just showing you, this is just showing you what's taking place as far as the sodium potassium pump. And you'll see here that it requires ATP, right? So it's a active transport, and we're going against the concentration gradient. So remember, if we have a channel here, which would be like a voltage gated or ligand gated channel, or a leak channel, which is always open, the gates are always open, what is going to happen with sodium? Sodium will move from outside to in. Potassium ions will move from inside to out. But in the sodium potassium pump, where there's energy required, we're pushing against. So you see here, we're taking 3 sodium and we're pushing it outward. We're taking 2 potassium and we're pushing them inward. That requires energy. It's going against the concentration gradient. This will enable the return of the resting membrane potential to its -70 millivolts. Okay, a little bit positive on the outside, a little bit negative on the inside. Okay, that's important to remember that and to get that. Okay, and let's see here, talk about this. Yeah, so the smooth and the cardiac muscle, I also have another little bit of information that I'm going to share with you all note-wise next week that I'll give you regarding that. And then we're going to come to, I want to discuss this right here. So I've gone over, in the lab, I've gone over all the muscles that you need to know other than the extrinsic eye muscles, right? So we didn't go over and look at the model regarding that. We'll do that next week, okay? But you'll see here that the naming and the actions of skeletal muscles, right? So the action is when a skeletal muscle contracts, what's going to take place? What's the action? So what kind of movement is going to be produced by its contraction? The shape of the muscle can contribute to the name of the muscle. The origin and insertion, sternocleidomastoid. How about the sternohyoid muscle, right? So these are, how about the stylohyoid muscle? You'll have origin and insertion in the name itself. Location can be an issue, and also direction of fibers. External and internal obliques, we definitely have the fibers are going in opposite directions, and you saw that in the lab, and you need to, again, review that and look at that in the lab and see how one side of the large torso model, you'll have the external obliques, then you'll have the internal obliques, and the fibers are going in different directions, okay? As far as origin and insertion, we've talked about these terms, right? Origin, more fixed attachment of the muscle. So this area really, when the muscle contracts, this area is going to be more of the anchor, and the insertion is really going to be where that muscle contracts, it's going to move a bone or bones, okay? They're parts of the body. The tendon, we know that a tendon attaches muscle to bone. We know that ligaments attach bone to bone. The aponeurosis, a wide flat tendon. So we saw aponeurosis on the posterior aspect of the torso, on the anterior aspect of the torso, as far as the abdominal, the rectus abdominis is covered by an aponeurosis. And how about the epicranial aponeurosis on the skull? So we have frontalis muscle, we have occipitalis muscle, or it can be known as the occipitofrontalis muscle, right? We call them separate ones, right? So frontal muscle, occipital muscle, epicranial aponeurosis connects the two muscles together. Flat, wide connective tissue. Terms that are used for range of motion. So flexors, extensors, abductors, adductors, adductors, rotators. On the superior aspect of the thigh, you'll see that I had mentioned about AD, adductor, longus, and magnus, right? I showed that they're more on the aspect of looking at the model there, more lateral on the model, okay? And so we'll actually more medial, sorry, more medial, apologize. More medial on the model, those adductors, adductors. Abductors would actually move a part of the body away from the midline, and adductors are moving toward the midline. Okay. Dorsiflexors, plantar flexors. So we looked at as far as pronators, so pronator teres. It even has the name of the action present within the muscle itself. Okay. Levators, so levator scapulae, right? So raising a part of the body, in particular levator scapulae, elevating the scapula, right? Depressors, depressor angulae. So levator, how about this? Levator labii superioris, right? Depressor angulae oris, depressor labii inferioris, right? So you see how the actions are involved in the naming of the muscle. Now, this is interesting. Prime movers are, or we would use the term agonist or antagonist. So if we have an agonist, it's bringing about an action, but an antagonist would move against, opposite of the agonist action. And a synergist would have musculature that's moving in this, doing the same type of action. So quadriceps can be synergists, hamstrings can be synergists. Quadricep and hamstring with each other would be antagonists because one produces one type of action, and the quadriceps and the hamstrings can produce another opposite action. Okay. Good. Okay. So yeah, so I'm not going to then move on any further. We have, as far as the PowerPoint's concerned, it's going to go over other muscles and just as a review. So let's press stop here.